Start now. So, hi everyone, my name is Harriet Chen. I'm a senior at Los Alamos High School, and today I will be discussing my project progress towards an expanded SAR of the C4 modified analogs of podophyllotoxin. And yeah. So first, I'm going to give everyone a brief introduction of what podophyllotoxin is and what it does, as well as previous studies on the natural product, our own studies, and our current work. So, Podophylltoxin is a natural product that is extracted from the wild mandrake plant, Podophyllum penteltum. Our ongoing project focuses on making modifications of podophylltoxin because previous literature demonstrates that 60% of FDA small molecule approved therapeutics in the last 40 years consist of natural products and their derivatives. But remarkably, synthetic derivatives of natural products and natural product inspired compounds outnumber natural products themselves in a 10-to-1 ratio. And this is because natural products are not optimized to treat human diseases, which basically marks a need for modifications of these molecules in order to actually get them to treat the diseases that we want. So going back to podophyltoxin, this natural product's mechanism of action is essentially binding to the alpha-beta heterodimeric interface of tubulin in order to inhibit polymerization. Previous studies have shown that the C4 position of podophyltoxin is responsible, or at least helps in regulating its mechanism of action. And this has been shown through previous studies, and because of this, there have been many modifications at the C4 position of podophyltoxin, and these compounds have then been sent to become FDA approved or in clinical trials. So clearly the C4 position plays an important role, and that is what my group wants to discover. Specifically, etoposide and teniposide, these are two FDA-approved small molecules of podophyltoxin. These show that essentially when you add a sugar group to the C4 position, which is in blue, you can change its mechanism of action to inhibition of DNA topoisomerase compared to podophyltoxin's original mechanism of action, which is inhibiting tubulin. Yeah, and this is just etoposide bound to DNA topoisomerase. And for my project, my group wanted to evaluate the structure-activity relationship of podophyltoxin, essentially relating the C4 position to cytotoxicity. And when looking at the previous literature, some researchers stated that increasing the C4 bulk decreases cytotoxicity, but others stated that there wasn't really a clear trend in these studies. So a lot of just differing statements. So because of that, we wanted to establish a systematic library of compounds that would allow us to really evaluate the SAR podophyltoxin. So as you can see from 2A to 2F, there is an increasing bulk at the C4 position. These are ester substituents. And as you can see, there's an increasing carbon for each one with the exception of phenylacetate. But phenylacetate is the bulkiest of all. So there's just an increasing bulk for this whole library of ester compounds. And from here, we wanted to test the biological activity of these compounds via MTT assays, flow cytometry, computational analysis, and tubulin polymerization assays. So I'm just going to quickly go over our biodata and then go on to what we're working on now. So for our MTT assays, which is essentially drugging cells with our compounds and seeing how well they killed cells, a trend that we observed is that as bulk increases, there's a decrease in antiproliferative activity. So the higher bars here indicate that more cells are alive after drugging. So as you can see, podophyltoxin was the most cytotoxic, and hexanoate was the least cytotoxic. And then phenylacetate was pretty unique because although it was the bulkiest, it did have a decrease in cytotoxicity compared to its less bulky compounds. And yeah, so then for our flow cytometry, which is essentially drugging our cells with our compounds and seeing what stage of the cell cycle our drugs kill our compounds in, we want to see majority of our cells being killed at the G2/M phase because we're assuming that our compounds are inhibiting tubulin polymerization. So, and since tubulin is a protein in the G2/M phase, we want to see cells being killed at that stage. So after doing the experiments, we can see that as you go from acetate through hexanoate, there is a decreasing amount of cells in the G2/M phase. But that same phenylacetate exception is being seen where it's actually there's actually more G2/M cells compared to like hexanoate. So that same exception is still there in both our cell assays. Additionally, we docked our compounds to tubulin and calculated the binding affinity, so how well our compounds are binding to tubulin, but we didn't really see any significant differences between all the binding affinities. Yeah. And we also did a tubulin polymerization assay, which consists of putting together tubulin, buffer, and our compounds together, and then measuring absorbance to see how well our compounds are inhibiting polymerizing tubulin. So the higher the absorbance, the more tubulin that is polymerizing. So as you can see there, our graph shows lower absorbances compared to Taxol, which increases polymerization, and our control. And this confirms that all of our compounds do inhibit tubulin polymerization, but there isn't really much significance between the levels of inhibition, but this assay allows us to confirm that there is inhibiting. So for our conclusions, in the cell assays, increasing bulk resulted in decrease in cytotoxicity. And this was, so this is our flow and our MTT assays. So those showed some sort of trend, but in addition to like the phenylacetate exception, but at the same time in our cell-free assays, which is the molecular docking and tubulin polymerization, we still aren't really able to establish some sort of relationship between our compounds and how well they inhibit it. And this leads us to question whether our compounds, or like increasing hydrophobicity of our compounds, is impacting how well they enter the cytosol and membrane of the cells, or if there are if there are other like non-specific ligand protein interactions.
Actually, by looking at this, okay.
Yeah, so because of that, we just want to increase our library of compounds to establish a more definitive SAR. So for our now instead of esters, we're also doing carbonates as well. So basically how the synthesis goes is that it's pretty simple and similar to our esters. It's just adding chloroformate with the substituent where the R is, and then DCM and triethylamine. It's around a 40-minute to 1 hour reaction. And as you can see, they look pretty similar to our ester compounds, except that there's just an oxygen as well here. So there's still an increasing trend that we're trying to observe.
Hold on for just a second. Nalini, can you please mute your
Got
mic?
it.
Thank you, David. Go ahead.
No problem. So I was just saying that we're trying to expand our library by adding carbonates as well. And they look pretty similar to the esters, but a different substituent with the same increasing bulk trend might help us give us a more definitive SAR, or like how well like podophyllotoxin, like the C4 position, works. And we also tried carbamates, and we're also adding carbamates to our library. And as you can see, we tried a lot of different conditions first for our carbamates because the synthesis is a little less known. So we tried like phenylchloroformates, which had no reaction, DSC, and isocyanates. And after that, we also tried diphosgene, and then added hexylamine. So because this showed like spot-to-spot conversion, we wanted to take that further. But after adding the hexylamine, which is like the second step to making the carbamates, we did not observe the intended product that we wanted via NMR, IR, and LCMS. And we suspect that this is the product here, the C3C4 endoolefin, that we suspect that is what we made. And this may be because of how strong the bases that we were using had an effect on the product. So for our next attempts, we wanted to use weaker bases such as pyridine, lutein, and DMAP. But eventually we were able to synthesize our carbamates. With the help of Ed, we made a nitrophenyl carbonate first, which is basically similar to the carbonates I talked to before. So we used nitrophenyl chloroformate with DCM pyridine and at a gram scale. And after that, we had this 4-nitrophenyl carbonate. And what's good about this nitrophenyl group is that it's a good leaving group. So when we added amine, we could see that conversion was instantaneously happening because our reaction solution turned yellow, which indicates that this nitrophenyl group was leaving and that the amine was creating the carbamate. Some byproducts that were as a result of our reaction was the nitrophenyl leaving group and urea. And some issues that we had with purification was that these byproducts the same RF as our intended product, so in hexane and ethyl acetate. So because of that, we had to switch our solvent systems to ether and hexane or MTBE and hexane. But after we figured that out, we were able to synthesize a large library of these carbamates. So we have our alkyl carbamates, which follow the same trend as the ester carbamates, the esters. The additions of terbutyl isopropyl, diethyl propargyl, and adamantium. And so we've synthesized those. And then we also have our cycloalkyl carbamates. So cyclopropyl, cyclobutyl, pentyl, cyclohexyl, benzyl. Benzyl isn't a cycloalkyl, but it does have a cyclic structure, which is something new that we want to observe with our podophyllotoxin analogs. And then next off, we have our heterocycloalkyl carbamates. So just adding variety to the bulk trends and the hydrophobicity trends that we want to analyze. So this week, what we accomplished is that we made almost all of our cycloalkyl carbamates, and we want to further study those because our biological data here, especially with our cell-free tubulin experiments, they show that small carbocyclic substituents at the C4 are well tolerated. So basically the cyclopropyl and the 4-methyl-pip were pretty good at inhibiting tubulin. We also did MTT assays for MCF7 breast cancer cells for some of our analogs. And currently they aren't really showing a clear trend, but we will be doing more MTT assays again, and also be doing different cell lines. So hopefully those will show a more clear trend. And we also need to wrap up our tubulin experiments as well. And then for our future work, again, just running more MTTs on colon cancer cell lines like HT29, HT116 cells, doing cell cycle analysis. So like before with our esters, we did that with the cell phases and G2/M phases. So we hope to see inhibition of tubulin or something different with our new analogs. We want to complete our tubulin polymerization assays and also do a computational analysis to tubulin for analogs. And I just want to acknowledge, thank you, Dr. New, for just giving us the opportunity to do this work. Thank you to my group members, and thank you to ASRP. Any questions?